Hello everyone, we are now going to go over the flow cytometry plots from a lymph node specimen from a 52-year-old male who is presenting with diffuse lymphadenopathy. So let's start with our first plot. Instead of the routine CD45 versus side scatter, this time we have provided CD19 versus side scatter. So here we see that majority of the cells are CD19 positive, which means they are B cells. And in a lymph node, we normally expect to see just the lymphocytes, but sometimes when they are performing the procedure, there is a chance that there is some contamination from the peripheral blood or some background granulocytes and monocytes. So the cells with high side scatter here that we see are those granulocytes, and maybe there are some monocytes, and there will also be some T cells which are negative for CD19. Now, our CD19 positive cells have high side scatter. Normally in lymphocyte population, we expect the side scatter to be very low because there is pretty much nothing in a normal lymphocyte to contribute for side scatter, not much complexity or granularity. But here we see a high side scatter, which indicates that these cells are larger. And now in kappa versus lambda plot, we see that majority or almost all of our B cells are lambda restricted. We do see some kappa positive background B cells that are in the dark red here that also have our CD19 positive here, but majority of our cells are lambda restricted and are neoplastic cells. And we see that in CD5 plot, these cells express CD5, the neoplastic B cells, and we can compare that with their background T cells, which are positive for CD5. They almost reach the intensity of CD5 of those cells. So they are neoplastic B cells express CD5. They are partially positive for CD38. They are negative for CD10, negative for CD200, and they express bright CD20. Again, here we see that they are partially positive for CD5. Same thing again, partially positive for CD38, negative for 10, and they express bright lambda surface light chain. So we have a large B cell lymphoma that is lambda restricted and expresses CD5. It is negative for CD10. So one thing we can say is that it is not phenotyping as though it is a germinal center B cell origin lymphoma. This is CD5 positive, 10 negative. So there could be a few things that can have this phenotype. One is diffuse large B cell lymphomas can express CD5. And those are usually associated with poor prognosis. That is, CD5 expression is associated with poor prognosis in diffuse large B cell lymphoma. The other thing is mantle cell lymphomas. They have the blastoid and pleomorphic variants. Those are large cells, and again, those will have high side scatter and will express CD5. But that's a tissue diagnosis. You need the stain cyclin D1 and SOX11 and FISH studies to confirm a mantle cell lymphoma. And CLL that transforms to diffuse large B cell lymphoma can express CD5, but CLL usually has a dimmer CD20. We see that here the CD20 is pretty bright, and also CLL has dim surface lambda, surface light chain here, or lambda light chains are pretty bright. So those are clues, but again, that's not completely reliable, but that's also a differential diagnosis. And the other one is prolymphocytic leukemias can have a picture like this. They can be CD5 positive, and those are large cells too. So these are the things that you have to keep in mind. So therefore, again, in this case, you cannot top line it as any specific diagnosis. You're just going to say CD5 positive B cell lymphoma, and in your comment, you will say that the cells have high side scatter, and this is indicative of a large cell phenomenon. So compare correlate with your tissue morphology to arrive at a final diagnosis. Thank you.